In 1967, a team of archaeologists working in the southern Ethiopian highlands opened a cave. Inside, buried face down in compacted sediment at roughly 2,000 meters above sea level, lay the skeleton of a man. No grave goods surrounded him. No pottery, no beads, no tools. Just a body, positioned deliberately in a way that hinted at a cultural practice nobody could interpret, in a chamber where the dry highland air had quietly preserved what heat would have destroyed anywhere lower. The cave was called Mota, after the village nearby. The man was given no name. His bones were logged, catalogued, and stored, and for nearly 5 decades, that was the end of the story. Nobody thought to extract his DNA. That is not an accusation. It is a statement about what was technically possible. Africa's heat destroys genetic material faster than almost any environment on Earth, and before 2015, no one had ever successfully recovered an ancient human genome from anywhere on the continent. The assumption was not that Ethiopian skeletons lacked interesting DNA. The assumption was that Africa's climate had already erased it. That assumption carried a cost nobody could calculate until it was finally tested. Because what was locked inside that skeleton would force geneticists to redraw the ancestry map of an entire continent. And what it implied about the bodies of the people who survived in those highlands, breathing thin air through genetic machinery found in no other population on Earth, is a story still being written. Here is the problem. Ethiopia poses to anyone studying human genetics in Africa. On a principal component analysis, the standard scatter plot geneticists use to map how populations relate to each other, most African groups fall into clusters that track with geography. West Africans here, Southern Africans there, East Africans distributed along a cline between them. The patterns are messy, as population genetics always is, but they are navigable. You can draw rough borders. You can see the logic. Ethiopians break that logic. On the same plot, they pull away from the rest of Africa and drift toward the Middle East and Southern Europe. Not subtly, not at the margins of statistical noise, by a distance large enough to make the graph look like it contains a misplaced data set. For decades, the explanation was a story of geography. Ethiopia sits at the Horn of Africa, barely 20 kilometers from the Arabian Peninsula across the Bab el-Mandeb Strait. Trade routes had connected the two shores since before recorded history. The Kingdom of Axum maintained ties with the Greco-Roman world from the 3rd century. The Persian prophet Mani listed Axum as one of the 4 great civilizations of his era, alongside China, Persia, and Rome. When early genetic studies in the late 1990s found that Ethiopian mitochondrial DNA contained nearly equal proportions of African-specific and Eurasian-specific lineages, researchers built a familiar narrative. Slow, bidirectional contact, merchants, diplomats, and intermarriage accumulating small genetic contributions over centuries. The drip of cultural exchange across a narrow strait. That narrative felt complete. It had the weight of centuries behind it. Christianity arrived in the 4th century. Semitic languages crossed the strait. The Kebra Nagast, Ethiopia's foundational royal text, traces the ruling dynasty to a union between Solomon and the Queen of Sheba. Every thread pointed to a country shaped by its proximity to the Near East, absorbing influence the way a coastline absorbs waves, gradually, predictably. But comfortable explanations discourage the question that actually matters. Not whether contact happened, but whether contact alone could produce what the data showed once someone finally measured it at scale. And the answer was no, not even close. The first signal that the slow contact model was wrong came from the linguistic map. Ethiopia is the only country in Africa that contains representatives of all 4 major language families found on the continent: Semitic, Cushitic, Omotic, and Nilotic. 4 language families in a country roughly twice the size of France. That level of linguistic compression does not emerge from gentle, sustained contact. It speaks to layered settlement, one population arriving on top of another, absorption and displacement repeating across millennia. But the genetic data made the case far more starkly. When researchers from the Wellcome Trust Sanger Institute in Cambridge generated genome-wide data from multiple Ethiopian populations and compared them side by side, the result was not a gradient. It was a partition. The Amhara, Semitic speakers from the highlands, carried 54% West Eurasian ancestry. The Oromo, Cushitic speakers spread across the Central Plateau, carried 51%. The Walaita and Somali, drawn from different linguistic branches, showed 43 and 44% respectively. And the Gumuz, a Nilo-Saharan speaking population living in the western lowlands of the same country, sometimes only a few hundred kilometers from highland Amhara communities, carried 0.7%, not 7%, not even 3%, less than 1%. That gap, from 54% to less than 1%, is not a gradient. It is a wall. And walls in population genetics do not form through trade. They form through replacement-level migration events that overwhelm one group and leave another untouched. Whatever introduced Eurasian DNA into the Amhara and Oromo either bypassed the Gumuz entirely, or the Gumuz resisted absorption in a way that kept their ancestry intact across 3,000 years. Either way, the slow diffusion model was finished. You cannot explain a 50-fold difference in non-African ancestry between neighboring populations through the gentle exchange of goods at a port. Something targeted happened, something that selected populations by language, by geography, by elevation, or by social structure, and left others entirely alone. The key to understanding what happened lay inside a particular bone. The petrous bone sits at the base of the skull, just behind the ear. It is the densest bone in the human body, a natural vault that shields DNA from the enzymatic degradation that destroys softer tissue. Ron Pinhasi, a co-lead on the study from University College Dublin, had spent years advocating for petrous bone sampling as a way to recover ancient DNA in warm climates where conventional extraction methods fail. With the Mota skeleton, the approach worked. The cave's high altitude meant cooler temperatures, the dry air limited microbial activity, and the petrous bone delivered. The resulting genome achieved 12.5 times coverage, enough resolution to reconstruct a full genetic profile with a contamination rate estimated between 0.29% and 1.26%. What emerged on the screen was, functionally, a snapshot of East Africa from before whatever had reshaped it. A 4,500-year-old genetic photograph with no Eurasian exposure. The team ran formal admixture tests using an F4 ratio estimate, the standard statistical tool for quantifying ancestry contributions. They searched the Mota genome for any trace of West Eurasian DNA. The answer was unambiguous. None. Zero signal. His genome was entirely East African. He carried 3 genetic variants associated with high-altitude adaptation found in modern Ethiopian highlanders. He lacked the derived alleles for light skin and eye color that characterize Eurasian populations. He was lactose intolerant, and his closest living genetic match was the Ari people of the southern Ethiopian highlands, the very region where his cave sits. Mota Man was, in a direct and verifiable sense, an ancestor of people walking the same landscape today. Except that his modern descendants now carry up to 25% Eurasian DNA that did not exist in his genome. Think about what that means. A population changed by a quarter of its total genetic composition in roughly 1,500 years. That is not gene flow. That is not gradual mixing at a trade crossroads. That is a mass migration event on a scale that physically replaced a significant fraction of the existing gene pool. The question is what arrived, and from where, and why. Something arrived after he was buried. Something enormous. If you are following this channel because you care about the moments when ancient DNA overturns established narratives, This is the kind of evidence we build every episode around. Subscribe, because the backflow event connects to patterns we are tracking across multiple pieces, and the picture is still forming. The team modeled the modern Ari genome as a two-way mixture, one component from Mota, one from an unknown Eurasian source. They then systematically tested every available ancient and modern population to find the closest match for that Eurasian component. Two candidates emerged above all others. The first was the genome of a 7,000-year-old farmer from the LBK culture excavated near Stuttgart, Germany, one of the earliest agricultural communities in Central Europe. The second was modern Sardinians, an island population known to preserve the genetic signature of early European farmers because Sardinia's isolation shielded them from the later Bronze Age migrations that reshaped the mainland. Both results pointed to the same ancestral population, the Neolithic farmers who left the Near East and Anatolia roughly 7,000 years ago, spread agriculture across Europe, and then, roughly 4,000 years later, somehow reversed course and poured back into the Horn of Africa. Andrea Manica, the study's senior author, put the scale directly. The wave of West Eurasian migration back into the Horn of Africa may have been equivalent to 30% of the population already living there. His co-author, Marcos Gallego Llorente, added that the genetic signature reached every corner of Africa. Not just Ethiopia. Not just East Africa. Everywhere. Because here is the part that overthrown the widest assumptions. Using Mota as an unmixed baseline and the Stuttgart farmer as the Eurasian reference, the team calculated Eurasian ancestry across the African continent. The Yoruba of Nigeria, the population most commonly used as a pure African reference in global genetic studies, carried 6 to 7% Eurasian ancestry. The Mbuti of the Congo rainforest, perhaps the most isolated population in Central Africa, carried 6%. These groups had been treated as baseline Africans in hundreds of published papers. Every model that assumed they represented pre-contact African genetic variation was now working with a warped ruler. Studies of migration timing, disease gene mapping, effective population size, any analysis that anchored itself to Yoruba or Mbuti as the African reference carried a Eurasian contamination nobody had accounted for. The correction rippled backward through a decade of published work. The mechanism of dispersal is itself telling. Eurasian genes did not spread across Africa through Eurasian people walking to West Africa. The Bantu expansion, a massive demographic wave beginning roughly 3,000 years ago as Bantu-speaking populations spread from West Africa southward and eastward, likely picked up and carried the Eurasian signal. Populations in East Africa absorbed the backflow DNA, and then the Bantu expansion, overlapping in time, redistributed it like a current-carrying sediment across the continent. There is a further twist embedded in this redistribution that rarely gets discussed. Eurasian populations carry roughly 2% Neanderthal DNA, a legacy of interbreeding that occurred after the original migration out of Africa. When the backflow carried Eurasian genomes back into Africa, it also carried Neanderthal sequences with them. Modern Ethiopians, with their roughly 50% Eurasian ancestry, carry approximately 1% Neanderthal DNA. The Yoruba and Mbuti, with their 6 to 7% Eurasian contribution, carry proportionally less, but still detectable amounts. Neanderthal DNA in Africa. That is not a sentence anyone would have written 20 years ago, but the data does not care about what sounds paradoxical. It tracks what happened. But the cause of the backflow itself remains genuinely unknown. No climatic event has been convincingly identified as a trigger. There is no evidence of a drought or famine pushing Near Eastern populations south. Archaeological evidence shows that Near Eastern crops, wheat, barley, lentils, arrived in East Africa at roughly the same time as the genetic signal, around 3,000 years ago, suggesting the migrants brought agricultural knowledge. The introduction of Ethio-Semitic languages into the Horn also aligns with this window. Linguists at the Max Planck Institute for Evolutionary Anthropology have noted that the Eurasian backflow theory matches models for how Semitic languages spread from the Near East into Ethiopia. But correlation is not causation. A root cause, a clear pressure that would explain why descendants of Anatolian Neolithic farmers suddenly moved en masse across thousands of kilometers into Africa, has not been found. One theory suggests that farmers followed the Nile upstream, looking for fertile land. It is geographically plausible, but numerically absurd. A migration equivalent to 30% of the receiving population does not happen because a few families wanted better soil. You need pressure, famine, war, collapse, something that makes staying worse than crossing a continent. A 2019 study published in Scientific Reports complicated the picture in a direction nobody expected. Researchers who separated the non-African component from Ethiopian genomes and compared it against a wide panel of ancient DNA found unexpected specificity. The Eurasian ancestry in Ethiopians did not simply match generic Anatolian Neolithic farmers. It showed particular affinity with populations from the Late Bronze Age Eastern Mediterranean, including groups connected to the disruptions that followed the fall of the Minoan civilization and the movements of the Sea Peoples, those enigmatic confederations whose raiding campaigns destabilized Egypt, toppled the Hittite Empire, and shattered the palace economies of the Eastern Mediterranean around 1200 BCE. The study even suggested, allegorically, that the mythological meeting of King Solomon and the Queen of Sheba might encode a folk memory of population movement between the Levant and the Horn during this turbulent period. The researchers themselves were careful. The deeply divergent African component that constitutes roughly 50% of most Ethiopian genomes creates statistical noise that makes it hard to distinguish between subtly different Eurasian sources, like Anatolian Neolithic versus Levantine Iron Age populations. But the signal was there, and if it holds under further testing, it would mean the Ethiopian genetic landscape was partly shaped by one of the most violent civilizational collapses in human history, refugees from a crumbling Bronze Age world finding a new foothold on the African continent. That debate remains open, and it connects to yet another unresolved argument in which Ethiopia plays a central role. The Horn of Africa is not only the site of the largest known return migration, it is also one of the two leading candidates for the original exit route of modern humans out of Africa roughly 60,000 years ago. Pagani and his collaborators sequenced 225 genomes from Egyptians and Ethiopians and estimated that the genetic split between Egyptians and non-Africans occurred around 55,000 years ago, while the Ethiopian split was deeper, around 65,000 years ago. Their results pointed toward a northern route through Egypt and Sinai. But the question is not settled, and other researchers have argued the data is inconclusive because modern populations may not represent ancient ones. Ethiopia, in other words, sits at the center of both directions of the most consequential human migrations in history, the exit and the return. What is not debated is the second dimension of Ethiopian genetic difference, one that has nothing to do with migration, and everything to do with survival at altitude. The Ethiopian highlands, where populations like the Amhara have lived for millennia, sit above 3,000 meters. At that elevation, each breath delivers roughly 40% less oxygen than at sea level. 3 human populations have lived at comparable altitudes long enough for natural selection to act: Tibetans on the Qinghai Tibet Plateau, Andean Quechua and Aymara in South America, and Ethiopian highlanders. Each group evolved a different solution to the same lethal problem. Tibetans carry variants in the EPS1 gene, a key regulator in the hypoxia sensing pathway, that keep their hemoglobin levels suppressed despite chronic oxygen deprivation. That EPS1 variant is remarkable in its own right. It likely entered the Tibetan gene pool through interbreeding with Denisovans, an extinct archaic human population, tens of thousands of years ago. Andean highlanders took the opposite approach, ramping up red blood cell production to increase hemoglobin, a strategy that compensates for thin air, but raises blood viscosity and the long-term risk of chronic mountain sickness and pulmonary hypertension. When researchers genotyped over 1 million SNP variants in high-altitude Amhara living at 3,200 meters and low-altitude Ari and Hamer populations living below 1,500 meters, they expected to find selection signals in the same genes. They found nothing. Neither EPAS1 nor EGLN1, the two loci most strongly associated with altitude adaptation in Tibet, showed any genome-wide signal of positive selection in Ethiopian highlanders. The researchers had appropriate statistical power to detect effects of the same size found in Tibetans. The genes simply were not under selection in these populations. Instead, the strongest signal appeared in BHLHE41, also known as DEC2, a gene involved in circadian rhythm regulation and the cellular response to hypoxia. PPARA, linked to fatty acid metabolism and energy regulation, also emerged as a candidate. These are genes that operate through entirely different biological pathways than the Tibetan solution. The physiological result is difficult to overstate. Amhara men living at 3,500 meters maintain hemoglobin concentrations and arterial oxygen saturation nearly identical to American men at sea level. They neither suppress hemoglobin like Tibetans nor overproduce it like Andeans. Their bodies appear to have found a third way, one so efficient that it mimics sea-level physiology in an environment where oxygen is scarce. Cynthia Beale, the biological anthropologist who first characterized this Ethiopian pattern in 2002, described it as the least studied of the three major high-altitude adaptations precisely because it looked, on the surface, like no adaptation at all. The numbers read as normal. But normal hemoglobin at 3,500 meters is not normal. It requires a biological explanation, and that explanation, 2 decades later, is still being assembled. This matters beyond academic curiosity. Understanding how Ethiopian highlanders maintain oxygen homeostasis through pathways that do not involve EPAS1 could open new therapeutic avenues for treating chronic hypoxia conditions at any altitude. The genes are different, the biochemistry is different. The medical implications could be entirely distinct from what Tibetan studies have produced. The Mota genome proved these adaptations were ancient. The 4,500-year-old skeleton already carried the altitude adaptation alleles found in modern Ethiopian highlanders. The trait predates the Eurasian backflow by at least 1,500 years. It is indigenous, evolved in situ, and it operates through genetic architecture that exists in no other human population on Earth. What makes Ethiopian genetics resist clean summary is that the country contains both patterns simultaneously. A massive external admixture event, one of the largest reverse migrations in recorded genetic history, layered on top of deep indigenous adaptation that predates any outside contact. The Gumus carry a genome that looks like what East Africa may have resembled before any of it happened. The Ari blacksmith caste, an endogamous group that married exclusively within its own community for generations, also carries virtually no Eurasian ancestry, possibly representing the genetic descendants of pre-admixture hunter-gatherers who were absorbed into a rigid social hierarchy rather than replaced. The internal diversity is staggering. Genetic variation within Ethiopia exceeds what is observed across geographic regions many times its size. Nilo-Saharan and Afro-Asiatic Ethiopian lineages appear to have diverged 11 to 16,000 years ago, long before any known Eurasian contact. The structure was ancient. The landscape, spanning lowland desert, rift valley, and highland plateau above 3,000 meters, carved genetic boundaries that held for millennia. And And today, in Addis Ababa, a city Menelik II founded in 1889 at more than 2,300 meters above sea level, the capital of the African Union, these layered histories live in the bodies of 11 million people. The new Ethiopian genome array, built with over 2.2 million variants specifically designed to capture African genetic diversity, was created in part from whole genome sequences drawn from these populations because researchers realized that the standard reference panels, built primarily from European and East Asian genomes, were blind to variants that matter for African health. Variants in drug metabolism, disease susceptibility, altitude response, traits that only make sense when you stop treating African as a single genetic category and start seeing the continent the way the DNA sees it, which is as the most genetically complex place on the planet, with Ethiopia sitting exactly where that complexity reaches its densest, most tangled, most human knot. 4,500 years ago, a man was buried face down in a highland cave, his genome belonging entirely to Africa. Today, his descendants carry the DNA of Bronze Age farmers, the altitude adaptations of a physiology found nowhere else, and the unresolved question of why, 3,000 years ago, the ancient world decided to reverse course and pour itself back into the place where humanity began.